All right, welcome everyone. This morning we're going to go on some really great lab tours with Dr. Adam Stieg. Today, Adam is going to take us with a team of people with Dr. Peng Ji and Dr. Michael Lake through CNSI, California NanoSystems Institute at UCLA. And we're going to see the Advanced Light Microscopy Lab, the Electron Microscopy Imaging Lab, and the Nano Pico Characterization Lab. So Adam, thank you so much for being here, and we welcome you, and we're very excited to listen and to see what you all have going on.
Great. Well, thanks so much, Caitlin, for the intro, and to all the organizers for putting this event together. It's interesting to do this type of thing remotely, but we've done it a few times, so hopefully we can pull it off smoothly. I'm going to switch over and share my screen to give you a little bit of background on what it is that we're going to see over the next hour. I'm going to start off just sort of giving you some introductory content, and then we are going to get up and walk around. So the next 20 minutes or so should be relatively clean and smooth because I'm just going to be talking to you through my computer. Then we are going to be transitioning to the three labs that Caitlin mentioned and doing a sort of walking tour and some live demonstrations. So hopefully the tech cooperates. We shall see what happens. I believe that questions are possible through the chat windows in YouTube feed. So if you do have burning questions, please send them through. And if we have some time, we'll try to address them. So to get the ball rolling on this, I'm just going to switch over here and go into a sort of short slide deck. And hopefully you can see me and see the screen at the same time. I'm basically just going to be talking about how it is that we use advanced technology to visualize the world around us. It's sort of the limits of resolution, pushing it down to the smallest of scales, because it's really important that we understand what it is we're measuring, what it is we're working with when we're doing nanoscience and technology. So, as Caitlin mentioned, I'm at the California NanoSystems Institute at UCLA. Many of you probably aren't aware, but there is a group of institutions that were founded just 20 years ago this year by former Governor Gray Davis. They're distributed throughout the UC system. And CNSI is one of them. They all focus on what we would consider to be grand challenge problems for society. CNSI is at UCLA and UC Santa Barbara, and we really focus on trying to develop and sort of foster the development of advanced technologies by, pushing innovation, basic science, but with an, sort of a problem-driven team science approach. And we do that in various ways. At this unique research institute here on the UCLA campus. I don't have time to get into too much of the detail, but what you can take from this is that we draw colleagues and partners from all across campus. It's quite a large building. We do research in a variety of areas. Nanoscience is pretty broad in general. It's certainly been expanding over the last few decades, but there's a lot of activity going on in all the areas that you could potentially imagine would be interesting to advanced science and technology. And then one of the other unique things that I won't mention very much today is that we do actually house a startup incubator for small companies. Currently, we have 25 companies in-house that are utilizing the research infrastructure and collaborating with our members here at CNSI that have proven to be quite successful. So what I'm going to mainly focus on today is how we provide advanced capabilities to enable discovery in cancer, nanomedicine, antibiotics, nano-environmental health and safety or sustainability, renewable energy, advanced computing technologies. And what we generally have to think about when we're developing these new types of materials is the fact that when we shrink things down to the nanometer dimensions, our traditional intuition that's sort of based off of the understanding of how the world around us works, sort of, this sort of Newtonian model of things, breaks down at a certain point and starts to become governed by what we generally consider to be quantum mechanics, where things are more based on probabilities and distributions of likelihood. And it's this sort of in-between space when we start to shrink down structures and objects and their behaviors become unpredictable, that really is the unique space where nanoscience can provide new capabilities, new properties, new materials that do exciting new things, but it also requires that we really are able to understand how structure and function are sort of inextricably linked. And so a lot of what we do here at CNSI and the labs that we're going to walk through today are facilities focused on characterization, because as much as we can make things at nanometer scales, we need to be able to understand what we actually made, because they're incredibly small. And that requires advanced technology. So there's a sort of delicate balance of, making and measuring that forms this synergistic loop where researchers are developing new devices, new materials, and people are developing advanced capabilities to measure them. And when we think about measuring, right, I like to use analogies as much as possible to make this accessible to people. And the analogy that I'm going to use here is how you and I measure the world around us, right? We utilize our senses. Generally, people think about the 5 different senses, and some of those senses give us information about different types of things, right? If we think about how we understand the physical structure of an object, we generally rely on our sense of sight, right? We also, if you could imagine, if you were blind and didn't have the luxury of sight, you could use your sense of touch to physically give you information about the structure of an object. Your nose and your tongue are sort of giving you information about the chemistry of the world around you. We generally don't taste things in the lab so much, so it's not going to give us, it's not a analogous technique to anything we're going to talk about today. And unlike bats or dolphins and other animals, we don't do a great job with echolocation, our sense of sound, to give us spatial and structural information. So when we think about how we measure physical objects, generally we're talking about our ideas of sight and our sense of touch. And I'm going to hit on that in a couple different ways, in the sense that some of the advanced microscopes we see are very much analogous to our sense of sight. And there's another class of microscope that's quite analogous to our sense of touch, right? So when we move through these techniques, we're going to think about the ways in which the microscope is measuring things. And depending on what we're measuring, right, if we want to measure a large object like a small animal, down to the smallest of objects like a molecule or an individual atom, there are different techniques that are available for us to do that. And we apply them based off of their applicability, but whether or not the sample itself can be analyzed, and whether or not the resolution requirements that we're hoping to achieve can actually be reached. And so if you look in the lower right corner, you see that our human eye, right, in this analogy of sight, can see things down to the ranges of, a few hundred microns for most of us. And as we start to want to see things that are smaller than that, we need to use more advanced techniques. And we're going to hit on a few of those today. We're going to be talking about optical microscopy, which is probably the most closely analogous to the sense of sight. Talk about electron microscopy, which is a sort of extension of that by playing with the electromagnetic spectrum. And then we're going to hit on scanning probe microscopy. And we're going to talk about them very briefly here and then go into the labs and actually see what that looks like. And so if we think about using light, right, like the way in which our eyeball is seeing something is that basically the light in this room, right, that's coming from the ceiling right now is bouncing off of me, off the walls, off the surfaces. It's being reflected, at least for me, what I can see, into my eyeball, right? The lenses of my eyes are focusing it onto my retina, which is my detector. That information translates into my computer, which is my brain. And then, my brain reconstructs a 3-dimensional image or structure of the world around me that's changing with time, right? And so the way that we think about looking with light is very much analogous to our eyeballs, right? Now, of course, we can look at how things are absorbed, how they're reflected, how they interact in various ways. But what we're mainly going to talk about today is how we take this analogy of your eyeball and turning it into a microscope. And the main portion here is that we add some additional lenses in between the light and our eyes to magnify objects in various ways. And so you're all pretty familiar, most likely, with a microscope image. And for many of you, you may have seen optical microscope images of something like on the upper left-hand corner here of a cell, right? And that was sort of status quo for many hundreds of years. I mean, microscopes have been around since the 1600s. But some of the major advances is what we're going to talk about now, which is that combining optical microscopes with chemistry, right, which is what I'm talking about, is adding these dyes we see here in the lower left-hand corner, basically allows us to paint the structures of cells, to allow us to differentiate between, for example, the nucleus, which you're seeing in sort of bluish purple, versus some of the structures that make up the physical architecture, what's called the cytoskeleton, the actin filaments or microtubules, other structures. And we use light using what's called fluorescence microscopy. Some of you may be familiar with sort of, ultraviolet, like black light paintings. We're basically using similar dyes to paint in a targeted way the structure of the cells such that we can not only see where things are, we can differentiate what they are. And that is a very sort of leap forward in the application of optical microscopes. Another sort of leap forward that's very common when we want to get to smaller dimensions, not just what are things, but we want to get better resolution, is sort of an extension of what some of you, again, may be familiar with, which is called a pinhole camera, right? And this has led to a class of techniques that's very broadly used called confocal microscopy. Basically, what you can see is that if you look at the top of this diagram on the right, you'll see that there's a thing labeled as a detector pinhole aperture. And what it's basically doing is it's adding a pinhole in between the lenses and the light path to basically filter out any light that's not in focus. So now instead of getting what we consider to be a wide field image here on the right-hand side, where you're seeing the full three-dimensional information of everything, you're creating slices, right? So that only the things in that plane are in focus, right? And so you obviously see a dramatic enhancement in resolution. But what you also see now is that if you scan that pinhole in space, you can now generate three-dimensional representations of an object like this, which is actually an artery, right? Looking at how cancer cells move through the vascular system, where you now can take individual slices and reconstruct them in 3 dimensions into a 3-dimensional image, where within every slice, right, we have a very, very nice high-resolution image where everything is in very clear focus, right? So confocal microscopy, the advent of this sort of technology, really pushed things forward quite a bit. Now, we've gone further than this nowadays. One of the sort of most recent breakthroughs resulted in a Nobel Prize in chemistry just a few years back. And they broke down a traditional law that was considered basically the diffraction limit, which said that the wavelength of the light, the size of the light that you're using, determines the resolution. And I don't have time to get into the details here, but basically this technique, one particular example called STED, which is what we're going to see a little bit of when we go to the lab next door, sort of manipulates light through what we would call destructive interference. So most of us are probably familiar with destructive or constructive interference. If you've ever been to the ocean and you see how waves interact, you visualize that physically. And basically, they can manipulate the structure of the light itself to give you a dramatic enhancement in the image. So you can see here on the left, for example, a confocal image versus a super resolution image on the right-hand side. And this has really overcome, hundreds of years worth of limitations on the resolution that you can achieve with light. And then most recently, which is really fun, is that people have reconsidered how these microscopes work, right? All of those measurements you just saw were done by scanning a laser, a single point, and measuring things point by point by point and reconstructing an image. That gives you very high resolution, but it actually slows things down quite a bit, such that processes and dynamics, things that are changing, become a bit tricky to measure. And so recently they've thought about ways to manipulate light into a sheet instead of just a point. And what that allows you to do is things like this, where we have now what's called a light sheet microscope. And this is actually a real-time image of a small fish called a zebrafish that we use quite often because it's actually transparent. And what you just saw there is actually a little light being shined into the zebrafish's eye. And now you're watching how the neuronal cascade, its nervous system, is receiving and transmitting that signal through its entire body in real time. So these types of new advances are done through instrument development, and they really allow us to push the limits of resolution and time with these advanced techniques. Now, I gave you a sort of example of optical microscopy to start with, because it's historically the most relevant. It's the sort of, the first microscopes that we had, but B, because it also makes the most sense to us because we think about how our eyes work. But there are still limits, and there have historically been limits with optical techniques because of the size of light. Pre-super resolution, you couldn't get down much more than a couple hundred nanometers using standard optical techniques. So the methodologies that people developed in the 50s were to say, well, if light's too big, what else do we have at our disposal? And so people developed a class of techniques called electron microscopy because we have a shorter wavelength. We can actually tune the wavelength depending on the energy of the electron, and we can push to higher and higher resolution. And so electron microscopy, if you were to have time to look at this diagram, looks quite similar. Instead of it being photons that we're measuring, now we're measuring electrons. There are some lenses that are here in this structure. There's a camera. So instead of the laser and some glass lenses, we have what are called magnetic lenses and some very fancy cameras that allow us to detect electrons. And what this allows us to do is to now generate, again, very high resolution structures of physical objects on the external surface. This is a technique called scanning electron microscopy, where on the left side, you can actually see the differentiation between different types of elements. So what you're seeing there is a map of the different elements on a sample, archaeological sample. And then on the right-hand side, you're actually seeing an image of a butterfly wing with very, very high resolution. That was the original technique that was developed. More recently, we've twisted that around a little bit and developed a class of techniques called transmission electron microscopy, which is what you're going to see when we go to the lab across the hall. We have a very strong specialization in transmission electron microscopy, and the transmission just means that the electron is moving through the material instead of bouncing off of the material. And it allows us to now achieve atomic resolution, right? Like individual atoms is what you're seeing here on this diagram of a nanoparticle, or we can see individual atomic structures of a virus, for example. So this is an example of an aqua retrovirus. We're actually imaging and working with some partners here on vaccine development for coronavirus, where again, we're looking at the atomic structure of the virus itself and understanding how its structure and its properties work together. You can see here sort of visualization of this virus, and if you were to zoom in, you can see the individual position of every individual atom. And this was another technique that won a Nobel Prize a few years ago for what's called cryo electron microscopy. Cryo meaning cryogenic. It's done at very cold temperatures. And then we are pushing new techniques forward, again, not only in cryo-electron microscopy on individual particles, but also in what's called tomography, where instead of just looking at a sample at one angle, we can tilt it and slice through it and generate now three-dimensional structures through various different types of materials using sort of looking at it from every different angle approach. So this is called tomography. This is a technique that's very much emerging here. And so the last one that we'll hit on is, I mentioned we're talking about these analogies of sight and touch. Those two, if you think about them, are really advanced technological versions of our sense of sight. They operate in a very similar way, right? You're firing a laser, photons at a sample, and you're looking at how it interacts with that sample at a distance. You are firing a coherent electron, an electron beam at a distance and understanding how those electrons interact with the sample. But the sample is over there. The detector is over here, right? I'm looking at something from far away. But if I'm blind, I don't have that luxury, right? So there's a different class of techniques that are more recently developed that uses a sort of tactile sense of interaction. And this is a class of techniques that we're going to call scanning probe microscopes, where we're physically now going to be scanning a nanoscale, let's say, for the sake of analogy, finger across a surface and feeling it like a blind person would touch an object or read Braille, right, to physically understand its structure. Now, the way in which we can achieve very high resolution, obviously we're not using fingers, my fingers are quite big, is that we use very, very sharp fingers, right? So we have these physical objects that we can manipulate and produce, where at the apex of this little needle here is an individual atom. And if we're measuring with an individual atom, we can now visualize structures at the atomic scale. And because we're using physical interaction, it actually allows us to manipulate matter atom by atom. And I'll give you a couple examples of that in a minute. Now, you may ask, what are we talking about here? Like, what are these interactions? And don't worry about the chemistry that maybe, some of you may be having shock. Oh, I don't want to look at chemistry because they didn't like it. And don't worry about the equations. The point here is just that if you think about two atoms, which are these red dots here, that are really far apart, if you forget about quantum mechanics, they are unaware of each other at all, unless they happen to be on a collision course. And what you're seeing right now is that as they get close enough together, right, those two atoms start to interact, because we know that the electron density is negatively charged, right? The nucleus has a positive charge, and you start to get sort of electrostatic and various different types of forces, positive and negative, pulling towards each other, right? Don't worry about this equation, but suffice it to say that there's an attractive force, right? If you get closer, you keep pushing those atoms together, you get a repulsive force that happens, right? And if you combine all those two together, for the physicists or the chemists in the room, you would see this type of a curve that you might be familiar with. But for the non-scientists or in the room, basically what you're talking about here is that you're measuring and you can detect the interactions between individual atoms at very, very close distances. This entire curve here extends only over a few nanometers. So what that means is we need to be able to bring this little finger very, very closely and very delicately to feel the surface of our sample. It allows us to do some things that we can't normally do, one of which is that it's able to give us what I would call true topography, right? So I mentioned before, a few minutes ago, tomography, where you can look at tilt, right? If you wanted to understand the topography of this mountain range, right, you can look down at it out the window of your airplane and sort of see the topography. You could go down a little bit and look out at the side and sort of get a sense of that topography. But if you really want to know the topography and it really matters, you probably have to, you know, at least in years past, walk through that mountain range and generate a topographic map. And this is incredibly important when you have materials like graphene, for example, which is another Nobel Prize winning material development, where the thickness of the material itself is sort of governing of its properties, right? None of these other techniques that I've mentioned so far give you that true information in the Z direction, right? About how thick and how rough materials are. And that really matters in terms of a lot of types of properties. So this microscope, because it's sort of moving through and on the surface of the physical object with nanoscale precision, gets you this information that you couldn't otherwise get. The other thing that you get is the fact that touching something gives you additional information. If you were here with me and I asked you to describe this object to me, you may say, well, it's translucent. You know something about its structure in that sense. It's orangey colored. You might have some sense of its optical properties or electronic properties. You may guess that it's made out of amber or glass, right? But unless you physically touch it, we're not going to taste it, but unless you touch it, you wouldn't know that it has a gelatinous property, right? So the physical properties of materials, are they hard? Are they soft? Are they sticky? Are they magnetic? Do they conduct electricity, right? All of these things are things that we measure through interaction, through touching. We don't measure them by looking at them from a distance. So this type of microscope allows you to do things that you can't otherwise do, right? So you can measure at very, very high resolution, pushing the upper limits on the upper left-hand corner of actually measuring the electron density inside of an individual orbital on a single atom, right? Identifying individual atoms, you know, measuring and identifying small molecules, manipulating individual atoms and molecules into structures like you see on this top band. This is more surface science and physics types of applications. Whereas if you're interested in biological systems, you can measure single strands of DNA where you can see the helical turns. You can measure the structure of proteins, of single cells, of cells moving and interacting to form tissues. So this particular class of techniques goes, as we like to say, from atoms to organisms and sort of everywhere in between. It's incredibly flexible and allows you to measure things that you couldn't otherwise measure with some of the other techniques. Now that said, right, there's no one technique that does it all, just like we have 5 senses, right, so that we can understand the world around us, right? The microscopes that we use in general approach things in different ways. We have a lot of systems that use analogous properties to the sense of smell or to the sense of touch or to the sense of hearing. And it's through this combination of all these techniques, which I don't have time to talk about today, and we certainly don't have time to tour, that we get a full understanding of the world and the materials we're developing at this smallest of scales, where things don't necessarily behave as we would predict them to behave because of this unique size scale that is known as the nanoscale. And so with that, I'd just like to thank, before we go off onto the tour, obviously the Art Sci Center, who's invited us to participate in this great event here at UCLA, particularly Professor Victoria Vesna, who I've known and collaborated with for almost 20 years now, Caitlin for the introduction, and Chella, who has been in the background helping coordinate. We're going to move in this sequence now into the Advanced Light Microscopy Lab. Director Laurent Bentalila, we're going to meet with Mike Lake, who's going to show us a demo of a confocal microscope. We'll then transition over to electron imaging, where Dr. Pengui will be showing us around a little bit. And I'm not sure if Hong is going to be here today, but the sort of leader of that effort is Professor Hong Zhou. And then we're going to come back into the room that I'm currently sitting in, and Mike and I will show you an atomic force microscope, this tactile sense of things. And so with that, I'm going to stop the screen share and switch devices. because I need to get onto my phone. And I actually, before I do that, I need to put on my mask because we are at UCLA on campus. I had my mask off. Hopefully you can still hear me because I was in this room alone. But now that we are going to move out and interact with the world, I am going to be putting my mask on. So let me switch over here. I'm going to mute this one, unmute this one, and start this one. And hopefully, if I can see in the chat window that we're good. If we're not good, somebody can let me know, but I think we're probably good. So what we're going to do now is we're going to take a walk down the hall. So we are currently, oops, it says no video yet. Okay, now good. Okay, so we are currently in the basement level, 3 levels below ground of the California NanoSystems Institute. And I always joke that despite the lack of a view of the ocean, this is prime real estate if the view that you want is of small things, because it's a very, very stable, specifically designed, noise-free environment for high-resolution imaging. And if we were to look down the hall, which I'll show you in a minute, we have a collection of laboratories down here that focus on different types of advanced imaging. So before we walk in, I think I can flip this around. We are currently entering the advanced light microscopy and spectroscopy facility. And we will walk through here. Now, these are shared facilities. They're open 24 hours a day, 365 days a year for people to come and use them. And before we jump inside the room with Mike, what you'll see here is that basically, how that room, the lights off, we won't open it, is that we have rooms with collections of advanced equipment. And there's Mike. We'll jump to him in a second. But what I wanted to show you really quickly is that we're going to be highlighting a bunch of fancy equipment like this that we purchase from commercial vendors. I also mentioned that sometimes we're developing technology. And so our mantra here at CNSI is that if you can't buy it, you build it. And an example of if you can't buy it, you build it is this particular microscope here, which is what I mentioned before, is a light sheet microscope. So you can see here, this looks like a science project, and it indeed was actually built by an undergraduate student who worked with the director of this facility, Dr. Bentalila. And the reason for that is that at the time, you couldn't buy this microscope. There was not a commercial producer of light sheet microscopes, and so we built them. All right, so we build microscopes when we can't buy them, and then we commercialize them and work with our industrial partners to make these advanced technologies that are being developed into something that's more generally accessible for the community. So I'm going to switch over, and Mike is here, and I think what we're going to do is I'm going to stop talking, and I think Mike is going to switch over, and hopefully this works. So I'm going to mute, and we will switch the screen over. Okay, so it sounds like there's a sound issue with Mike, which is fine. We'll just use my phone here and use his screen. So where we are right now is basically in the light microscopy facility, and Mike is sitting at the control station of a confocal super resolution microscope, which is over here on the right. So hopefully, Mike, they can't hear you unless you talk through here. So I'll just hold it up here, and but they are using your video feed.
Okay, great. Well, basically what we're seeing here, you can see the screen updating, is actually a live image of one of the slides we just imaged. It's a slice of a worm. And you know what we can emphasize is here at the control station, we can control things like the field of view, the brightness, for example, whether we're overexposing or underexposing the colors, all of these manipulations. We can also move around the sample. When we get into what we're looking at, and going back to the theme Adam was talking about, looking at what we normally see through a microscope. The biggest innovation with confocal, it's hard to appreciate how revolutionary it is that with standard confocal standard microscopy, you're only able to see one field of view. This is transmission mode. And if you look, normally it would be black and white. And this is what you get when you scan up and down through the object, is you can see at all stages, you can look, but you have a blurry version of the image. So with this microscope, we're actually able to do two things. One, we're able to use color. So by collecting two different colors from the sample, we're able to separate out different components. We're also have the advantage compared to, for example, the atomic force microscope, that we can actually look directly through the sample and actually look slice by slice inside, almost as if we have X-ray vision. And so if you look at this sample, as I move down the Z-stack, unlike our previous transmission image, which you'll still see there in gray, as we step through, we only see one slice at a time. And you can see the kind of fine details, especially in the green, this fiber network right in the middle of this dense patch that we would never be able to identify, or these hollow cavities that don't show up in either color that also help us figure out exactly what's in the three-dimensional makeup. You can also see that we have two components in these organ systems, whereas in the red color, we have open spaces, and in the green, we have bright circles filling those spaces. And so we've successfully been able to separate two different biological components by the utilization of color. So that's a big advantage in light microscopy, is most things are transparent to light, and we're able to separate the components by color, helping us to dissect the biological components that we care about from the things that we either want to compare to or don't want to see.
Okay, so if we can switch back over to the live feed on my phone, just also give you a sense of what this looks like. So the microscope itself, right, this is for those of you who've seen these before, it looks like a microscope in the sense that, we've got some eyepieces that we look into to align things and to see what's going on. But we also have a bunch of other stuff going on here. One is that we have some really fancy cameras, right? And two is that we have lasers, right? So instead of just using a light like the light source, a white light source like we're traditionally used to, we use very fancy lasers that can control the way in which and how we illuminate or stimulate the sample with these photons that we're measuring with. And then it's all controlled, of course, by a complicated control system with some fancy computers that somebody like Mike or one of our many users that come here to use the facility can use to acquire data. I also mentioned before, that we're in a very quiet environment. This is actually what's called a vibration isolation table. It's actually floating. What you're hearing right there is me touching it, pushing it up and down, and then it's absorbing vibration, because anyone who's taken a picture of something that's moving knows that vibration or shaking is a problem. And then the last thing, just because we won't have time, I'll just mention, is that this here is actually an atomic force microscope that does mount. If we move it, it actually does mount right on here. Now, we didn't have time to do it for today, but this allows us to do simultaneous optics and atomic force microscopy, these scanning probe measurements, where we can look and see how physical structures respond to force, right? If you push on a cell or on an organism, for example. This is what we call correlative microscopy. It's a sort of emerging area of microscopy where we're trying to get multiple different types of data on the same sample at the same time, right? To be able to do various different classes of techniques. And we try to combine techniques together to give us a better view of the world in which we're measuring. So with that, I'm looking at the clock. We need to rotate over. So just also say thanks to Mike. You're going to see Mike again over at the atomic force microscope in a few minutes. Right now, we're going to walk across the hall and go see the Electron Imaging Center. One thing that I'll mention while I'm walking is that microscope that we were just looking at was actually an example of where a construction project turned into a commercial product. So if you actually look, I guess the poster that we had has been taken down. But that system was actually serial number, I think, 002. It was the first one in the west coast of the US that was supported by a funding from the National Science Foundation through their what's called a Major Research Instrumentation Program. to develop super resolution microscopes into a commercial product. So that was the gentleman, Stefan Hell, one of the Nobel Prize awardees, worked with the team here to develop this class of techniques, which also partnered with Leica, the company that builds it, to commercialize it. So that's another nice example of how technology gets developed and pushed forward. So where we're at now is let me see if I can get the logo here. We are entering the Electron Imaging Center for Nano Machines, and we are walking into the room to see what we're going to talk about here, which is in a very advanced transmission electron microscope that works at cryogenic conditions. So I said, remember, cryo is cold. The system is basically operating at the temperature of liquid nitrogen. And what this particular microscope allows us to do is to sort of look at structural biology, the structure of physical objects like the viruses that you saw, proteins, etc., one at a time and with incredibly high resolution. And so with that, I'm going to turn it over to Dr. Pengui, who's going to give us a little bit of a tour about the transmission electron microscope and its unique capabilities.
Thank you, Adam. I'm Peng for the EICN facility here, and I'm glad to show you guys what we have for the electron microscopy. Actually, this is a live running system, means we actually are running a product on it automatically without any human intervention. So as you can hear, if I get close, you can hear the movement of the stage. By the way, I'm very grateful for the talk that Adam gave earlier today, in which all the 5 senses of science actually come together to get you a full picture. So I used to think in terms of resolution because I'm a probably EM person. They always ask people, can you see an atom, or how many angstroms can you see for your resolution? Just like if you have a Porsche, you always ask people, how many seconds does your car take to go from 0 to 60 miles per age? So this one probably is the Porsche of electron microscopy. So it's one of the first similar units. That's the first mass production unit that has been in the both the United States and the world. And as you can see here, we have a monitoring station that is currently having shifts in pictures that are being taken. I think it's a viral particle from a NIH-sponsored consortium user, but I can't say what it is because we are serving the user. And as you can also see, the end product of this electron microscopy will be things like a 3D model of a microscopic object. In this case, it is a bacterial puncturing machine that tries to find the competing bacteria and kill that bacteria, which can also be utilized to make a very narrow spectrum of antibacterial thing. So to get to this kind of image, we also use very small tools to fixate your sample. This is our micro toolbox. We have all the tweezers. We put the things in the grid that doesn't even show up well on the phone camera. And it's actually made of these clip rings and these base grids, and we put those in the cassette, and the cassette is loaded into the cup just like this one. And this one is put into the electron microscope with an automatic reloading robot on its side. So this is a robot that takes 12 grids at a time and put them into the microscope one by one if you want to look at them. And the microscope is automatically cooled by the cryogen, as Adam talked about. This is liquid nitrogen. And this is the 300,000 volts of electricity applied to the tip of the electron source. As you can also see, we have a monochromator here that's trying to pass the electron through and remove whatever color of electron, quote unquote color, that you don't really want. This is a better view from this side. This one is a vacuum pump that serves the projection chamber, and this is the camera. The camera is mounted on the far side of this monochromator.
And when Peng says camera, this is not your traditional camera. So this camera can directly collect single electrons, right, which is a very, very fancy camera that allows us to push the sensitivity of the microscope and allows us to go at very, very high speeds to achieve these really high levels of resolution. So this microscope is basically, as Peng said, the Porsche or the Ferrari of this type of technology. You can see that it's built into its own room, right? You sort of, as we've been looking around, you can see it's quite large, right? It's built into a customized room in the basement, right? So that it can produce very, very quiet environment to do these types of measurements. But also it's quite tall. And part of the reason that it's quite tall, I think it's probably 12 feet tall, is the fact that that 300,000 volts that Peng mentioned of electrons those electrons are very high energy. They get accelerated down through that column, it's called, where there are lenses to focus them, because the energy defines the wavelength of that electron, right? So to get higher and higher resolution, you need a smaller wavelength. You need a more energetic electron that needs to be focused, and we focus it down from the, basically through this large column as we go. So this is a really nice example of, the type of infrastructure that the CNSI provides that most institutions or, certainly individual faculty members or small companies couldn't afford. And we have collections of these microscopes. We don't only have Ferraris, we have Fords as well, right? That I guess I shouldn't say that having just watched Ford versus Ferrari, because there are some fast Fords as well. But nevertheless, we do have different levels that apply to different areas. This is for structural biology, as I mentioned. Next door, we have a sort of the sister of the Titan Cryos, which is the Titan Scanning Transmission Electron Microscope, more for material science. So we have a whole suite of different technologies that are available for people to come and use, and we train people on how to use them. We have thousands of people who come in every year to use all these different technologies. So we will thank Peng real quick. I think he's looking at me with his camera. This is a great shot. And we're going to see him again in a minute. He's going to run upstairs to our data center, new data center, just so that we can show you what and how we deal with all the data that's coming out of these new microscopes. So thanks, Peng. We'll see you in a couple minutes. And now we're going to walk across the hall to our last stop. And I guess I should point out, it's quite nice, right, that all of these labs are in the same building, right? You generally don't find this. So this is the Nano and Pico Characterization Lab. It's one of the many labs that we have here. You see that there's a whole hallway of facilities just on the basement level. Inside each of these rooms are many different microscopes. And then, above us is a whole clean room that is for fabrication. So there's a lot of stuff going on. We're going to go into this lab and show you how this scanning probe microscope works. And now you will recognize the gentleman sitting here, which is Mike again. And I think what we'll do is basically switch over the screen share here because this computer is also logged in. And you can do your screen share from that particular microscope. Minimize that maybe. And then we should be able to see what's happening here. So I'll give a quick intro, and then Mike can walk you through what we're looking at on the screen before we switch up to Peng. So actually, can we pin my view right now really quickly just so I can show them the microscope because I'm seeing the interface. Actually, I guess that's probably because he's sharing. Can you stop the share? Yeah, stop the share. Okay. So this is an atomic force microscope or a scanning probe microscope. I'm not going to have time to get into the details of it, but you can imagine that it's also very sensitive. It's sort of like a record player, right? In the sense that instead of turning the grooves on a record into sound, it's turning the bumps on the surface into images. And so there's a small sample down there with a tiny little finger that's touching down and scanning across the sample. We'll actually see that finger in a second. It's called a cantilever. But the whole microscope is, again, floating on one of those optical tables that stabilizes it. It's inside of an enclosure to keep out electromagnetic and acoustic noise. And then once we get our tip onto the surface, maybe share the screen now, I can talk over it. Then you'll basically see that the tip is now going to be scanning on the surface. And what you're going to see now from the screen of the microscope is there's a little, looks like a paddle. Basically, that paddle is what we call our probe. It's a cantilever. It looks like a diving board, basically. But on the bottom side of that, underneath those crosshairs, is a very, very sharp tip, that little sharp needle that we had. And so that tip is now touching very, very delicately on the surface of a membrane that we're going to be measuring. And I think Mike is going to show you there. And what we're seeing now, if you look at the screen, is basically that the tip is scanning back and forth. I mentioned before that most of these high resolution techniques scan the tip back and forth and collect images point by point. And what you see here in the image is that as it comes down, tick, 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 you'll see that there is an image being generated. I don't know if you can see the cursor probably, but these images are composed of lines. If you see this horizontal line, it's composed of lines of the tip scanning back and forth, back and forth, back and forth. Where the color, right, is just a representation now of the height, right? You can pick your favorite color, right? It doesn't really matter what color you want because the color is not real. It's just a representation of height, right? And at every individual point, right, it's plotting the position. It creates that into a sort of pseudo three-dimensional image, right? And this tip now is moving across the surface, pushing very, very delicately. I don't know, Mike, can you see what's the force?
25 piconewtons.
Okay, so the amount of force we're applying here is 25 piconewtons. And for those of you who don't remember your Greek prefixes, it goes, milli, micro, nano, pico, which is why this is also called the Nano and Pico Characterization Lab, because some of the things we measure are smaller than nano. And so now it's tapping up and down on this surface very, very delicately. And this is actually what we're seeing on the screen over here is the actual individual taps of the tip on the surface to basically scan this surface and generate an image. So what you see here, right, is an image of this filter membrane. And the reason that we show you this particular sample is because of various reasons. One is that we're getting real topographic information, first of all. I talked about that before. These lines show that the bumps on the surface up and down are about 50 nanometers deep, right? There's not a good way to measure that quantitatively, where the numbers are real, with any of the other techniques. Similarly, this is a non-conductive sort of insulating membrane. If you were to put this into an electron microscope, right, you would not get any data out of it because it's not conductive. There are some sample requirements for an electron microscope that would limit our ability to visualize these structures. Even though the size is within the right range, the sample itself is not compatible. Similarly, if you put it into an optical microscope, it's opaque. Right, so the advanced stuff that Mike was showing before generally relies on the sample being transparent. Right, this is an opaque sample, so it doesn't work so well. And then, independent of that, even though these features in the XY plane are quite small, in the Z dimension, right, we wouldn't really have the ability to measure that information. So structurally, it's interesting. Now, I mentioned in the talk, in the brief intro, that not only can you look at structure, right, you can also, because you're touching it, measure things about Is it soft? Is it sticky, right? What are its mechanical properties? So Mike's going to click on an image right now, which is now showing you the distribution in the mechanical properties. So this is called the modulus, or the Young's modulus of the material. It basically says that we can monitor how the tip is interacting with the sample, right, and see in real time how much it's deforming. If we increase the force, the sample will deform more because it's not infinitely stiff, right? If we change it, if it's less, it would deform less. And we can now get maps of the local mechanical properties. Similarly, because we're touching it, right? Like if you touch anything, there is some attractive force there, right? And if we look at the adhesion channel, which is this channel, you can see that certain regions are softer versus others that are stickier. What I mean by that is that when the tip pulls off the surface, it's being grabbed onto, kind of like a fish, right? When you catch a fish and it grabs the lure and swims away from you, it's pulling the fishing rod. The same thing is happening here, right? When there's an attractive force, when there's a binding event, a bond formed, it pulls on the tip. And that's what we're monitoring here. And so there's a whole bunch of channels, which you can sort of see on the left. There's a sort of vertical panel of 8 different channels. You can click on whichever ones you want, Mike. This is what's called the dissipation channel, how much energy is being dissipated. There's a channel called deformation, where we can look at how it's deformed. And you see that a lot of them are similar because the structures are it's the same type of structure, but it allows us to map out these mechanical properties of a material, again, with very, very high resolution. So this type of microscope gives us access to things that we couldn't otherwise measure, especially with spatial precision. And can you stop the share real quick? Especially with the spatial information that we're talking about, right? So there are ways to measure stiffness or stickiness or conductivity or magnetism, but they don't provide you the spatial information that we're looking for. So by combining a microscope with what this class of techniques is generally called spectroscopy, we're doing, again, different modes of imaging and measurement at the same time while retaining our ability to resolve that information at nanoscale dimensions, right? So with that, unless Mike has anything else he would like to say, I'd just like to thank Mike, who's sitting here driving the bus for me this morning. And then I think if Peng is ready, we are going to jump back upstairs to the new data center. And Peng is going to talk briefly about how it is that we handle all of the data that comes out of these microscopes. And to give you a sense of what I'm talking about, one of those electron microscopy reconstructions that you saw generally takes a few 100,000 individual images, right? That light sheet microscope I showed you before can collect tens to hundreds of thousands of frames per second, right? So you've got an immense amount of data and a pipeline of data that needs to be processed, stored, manipulated in real time in many cases, or even after the fact, that we have to be able to handle. So we have unique infrastructure, not just microscopes, like rooms like this, right, filled with microscopes, but we also have the IT backbone to take the data off these systems up to where Peng is in the data center, and then the computing capabilities to be able to process the data to draw meaningful insight from that information. So hopefully Peng is there. And if he is there, then we should switch over to him. And he is going to give us a brief introduction. Yes, he's there. I will stop talking. I'm going to run up and meet Peng to wrap it up in a couple minutes.
Thank you, Adam. This is our new computing space. There's a human factor and a computer factor. This side is the human factor. So I'm sorry for the bobcat fire that actually made the very sunny California sun look like a evening time. And this is our human factor of the computing infrastructure, where people tries to understand the structures that are solved by the computers in the computing factor. We have state-of-art 3D modeling devices like the virtual reality headsets. We have a visitor office that allows prestige scientists and maybe artists who have the luxury of a sabbatical to come and join us for a enjoyable time. Those few offices, this is the office for Dr. Hong Zhou. Towards the end, also the COVID-19 actually was heavy on us too, and made this very lively place a little bit quiet. But we are just trying to find different ways to cope with it. And this is our paper writing station. For example, I developed a way to smile with my eyes only, given that my mouth is covered by the mask. This is the computer factor of the computing infrastructure. So I'm waiting for Adam to come upstairs to give the final conclusion, and then I will walk to the computer factor to show you those computers. I think Adam maybe at the door.
Yes. I am at the door, but I can't get in. We upgraded.
Yeah. I'm walking the other way around.
Okay. So in any case, I know that we're going to wrap up here in a minute. I think Peng is going to walk down and show you if we have any last minute while I sort of wrap things up. You want to go that way? In? Okay. So with that, I'll give you the other view of the Institute, right? So Peng, we only have 1 minute, so maybe just go down and go open the door. So the Institute is quite large. It's 190,000 square feet. It's actually built over the top of a parking structure here at UCLA. But there's a lot of different activities that happen inside of here. There are research laboratories. There, as I mentioned, are startup companies, our technology centers, which is some of what we've highlighted this morning. All sort of focused, again, on bringing teams of scientists together from different disciplines. Actually, the Art Science Center, Art Sci Gallery, is across the way over there. So it's really about bringing people together from different disciplines to solve problems in unique ways. Many cases focused on, how it is that nanoscience and the unique properties of the world at these dimensions allow us to do things we couldn't otherwise do. And so I know it's a bit loud in here. And I don't know how much time we've got, but basically what we're going to be Peng is here looking at our new data center, which we have just commissioned in the last few weeks, because we, as I said, need new infrastructure to be able to handle all the types of data that are coming out of these microscopes. So we have a very nice sort of dedicated environment filled with high density storage and computing nodes, both traditional computing nodes as well as GPUs for different types of applications. So I don't think, I'm looking at the clock, I think we're pretty much out of time. For those of you who like computers, you'll recognize racks of servers, etc. It gets a little bit loud in here, so I'll walk through and then back out. But I'd just like to wrap it up, sort of thanking all of you for your attention over the last hour of this sort of brief nano view of the nanoscale activity going on here at CNSI. And I'm happy, obviously, to answer any questions that anyone has in the future. You can find me on the web. Obviously, the organizers know where to find me. Happy to answer any questions. So I'm going to get out of the loud room, and I will call it a wrap there. Unless there's something else, Caitlin, that you need, let me know.
No, I just want to thank you all so much. Thank you, Ping. Thank you, Mike. And thank you so much, Adam, for taking the time to talk with us today. I love all the anecdotes, and it's just so amazing to get to see all these amazing microscopes and Porsches of microscopes so close up. So thank you very much.
Yeah, it's a pleasure. Thank you.